Welcome to today's Explainer. We are diving into our source materials to uncover how scientists actually read the active instruction manual of life. And honestly, more importantly, how they make absolutely sure the data they're reading is flawless. Have you ever wondered how we figure out exactly what goes wrong inside a diseased cell compared to a healthy one? Well, the answer lies in the massive amounts of data generated by genetic sequencing. But here's the catch. Dealing with millions of lines of genetic code isn't just about reading it. It is entirely about quality control. So let's explore this incredible process together. To make sense of all this, our sources lay out a really clear roadmap for us today. We're going to hit 6 key areas: the instruction manual of life, the bioinformatics assembly line, grading the raw data, spotting factory defects, cleaning and aligning, and finally visualizing the final picture.
Okay, kicking things off with Section 1, the Instruction Manual of Life.
So if you've ever wondered how we know what makes a diseased cell different from a healthy one, the secret is a technique called RNA sequencing, or RNA-seq for short. You see, every single cell in your body contains the exact same DNA, right? But a brain cell obviously acts very differently from a heart cell. Why is that? Well, it all comes down to gene expression. As our provided texts highlight, RNA-seq basically captures a real-time snapshot of this activity. It tells us exactly which genes are turned on in a specific cell at a specific moment and just how intensely they're working. Our source material offers a fantastic analogy to help visualize this. Think of DNA like a massive, heavy reference cookbook locked safely away in a library. It has every recipe for your entire body. RNA, on the other hand, is just a single, specific recipe page that a cell has photocopied so it can actively bake a protein right now in the kitchen. When we perform RNA-seq, we are essentially sweeping up millions of these tiny genetic photocopies from the kitchen floor to see exactly what recipes the cell is cooking up at that exact moment. Moving right along to Section 2, the bioinformatics assembly line. Our research outlines how we actually handle this mountain of data using guided software pipelines, like a tool called RNA Flow. I want you to think of these tools as our very own high-tech factory assembly line. We start by downloading raw shipments of data, which arrive in these huge packages called FASTQ files. From there, we meticulously inspect them for quality, we map or align them back to that master cookbook genome, and finally, we count and mathematically analyze them. And as the documentation stresses, every single step relies entirely on the success of the step right before it. This brings us to a really crucial point made by the researchers. It's basically the golden rule of bioinformatics: garbage in, garbage out. Quality control is hands down the most important step you can take. If your raw sequencing data has hidden problems, literally every downstream result you get is compromised. Skipping the quality control step could lead to weeks of wasted time, a retracted scientific paper, or worst of all, a false medical discovery. That makes our inspection phase the absolute most vital part of the entire factory. Which leads us perfectly into Section 3, grading the raw data. To prevent a total data disaster, our sources point us to a grading system based on something called Phred scores. You know, our sequencing machines are amazing, but they aren't perfect. Sometimes they make mistakes when reading those genetic letters. So the machine assigns a confidence grade, or a Phred score, to every single letter it reads. A score of Q10 means there's a 1 in 10 chance the letter is just flat out wrong. Q20 is a 1 in 100 chance. But what we really want to see are scores of Q30. That means there's only a 1 in 1,000 chance of an error, which gives us a fantastic 99.9% accuracy. Now, an inspection tool called FastQC visualizes these grades using a box and whisker plot. Picture a chart where you want to see all your data floating safely high up in the green zone across the whole sequence. That means excellent quality. If things start dipping down into the yellow or red zones, we've got a problem. Although the sources do note one exception. A slight dip in quality at the very end of a sequence read is actually a totally normal quirk of how the chemistry works in the sequencing machines. So don't panic if you see that tiny drop-off at the tail end. Let's jump into Section 4, spotting factory defects. Even if you have high overall scores, the documentation is quick to remind us that we have to play detective. We need to differentiate between an actual factory defect and a normal biological reality. For instance, finding synthetic barcodes, which the pros call adapters, left on the DNA, that's a technical error. We have to fix that. But what if you find millions of duplicate copies of the exact same sequence? Well, in RNA-seq, that's often totally normal biology. Remember, a highly active gene naturally pumps out thousands of identical photocopies, so duplicates aren't always bad news. This actually brings up a fascinating question posed by the researchers. Are all FastQC failures actually failures? The thing is, the FastQC software has no idea what kind of specific biology experiment you're running. The text gives a great example. Say you're running a specific chemistry test called bisulfite sequencing, which intentionally converts the DNA letter C into a T to study how genes are controlled. Your FastQC report is going to show a terrifying massive loss of the letter C and flag it as a catastrophic failure. But for you, it's proof your chemical experiment actually worked perfectly. Context is literally everything here. That being said, there are definitely true defects we have to watch out for. If you see a rising curve of adapter content at the end of your reads, it means those synthetic shipping tags are still attached. Not good. If you see a bimodal curve, basically a double-humped curve, your factory has a genuine defect. It strongly hints that some other organism's DNA accidentally contaminated your sample. And of course, if you have widespread quality scores crashing down into the red zone, the sequencing machine itself might have just malfunctioned. Any of these issues need to be addressed immediately. All right, Section 5, Cleaning and Aligning. According to the materials, once we know what's wrong, we send the data through a series of specialized software tools to fix it. First up, a tool like FastP acts like a pair of factory scissors. It trims off those synthetic adapter barcodes and cuts away any low-quality red zone bases. Next, we bring in an aligner, like HisSat2. Think of this tool as an expert puzzle solver. It takes our millions of tiny, cleaned-up RNA puzzle pieces and maps them against that massive master reference cookbook to find exactly where they belong. Finally, a tool called Feature Counts acts as our accountant, tallying up the final score of exactly how many puzzle pieces landed on every single gene. So, how do we know if our puzzle solver actually did a good job? We look for a 90% or higher overall aligning rate. If HisSat2 spits out a 90%, you can celebrate. It means almost all of your sequence pieces found their exact proper home in the master instruction manual. But if that number drops below 70%, yeah, it's time to pause the factory and investigate for contamination or weird errors. And finally, Section 6, Visualizing the Final Picture. To actually make sense of all this math, our sources highlight statistical packages like dseq2, which generate some pretty famous visualizations. First is the volcano plot. I love this one because it visualizes all your genes simultaneously. The x-axis shows the fold change, meaning how much a gene's activity went up or down. The y-axis shows the statistical significance, basically how confident we are that this isn't just a random fluke. Visually, it looks exactly like an eruption of dots. The highest dots shooting out to the far right and far left, those represent the genes that changed the most significantly between your healthy and diseased samples. Those specific dots are your prime biological targets. Next, we can also generate what's called a heat map. Imagine a grid where every row is a single gene and every column is a different sample. Blocks of dark red instantly show us genes that are running incredibly hot and active, while deep blues shows genes that are turned off or running cold. This is amazing because it instantly allows us to see patterns, clusters of genes that are acting identically within our treatment groups, which helps reveal hidden biological pathways. Finally, the data gives us the ultimate big picture, the PCA plot. This plot is wild because it takes the expression data of thousands of genes and compresses it all down into a simple 2D map. Here, each single dot represents an entire sample. If your healthy sample dots cluster tightly together on one side of the map and your diseased sample dots cluster tightly together way over on the other side, you can breathe a massive sigh of relief. You know your biological experiment was a complete success because those two conditions are genuinely, fundamentally different. So, what does this all mean for us at the end of the day? Well, as our sources have shown, quality control isn't just some boring chore where we delete bad data. It is the absolute foundation of biological truth. If we can build a perfect, well-inspected factory based on the principles we've talked about today, we are genuinely one step closer to unraveling the mysteries of disease itself. As these bioinformatics pipelines become fully automated, what hidden cures will we find waiting in the vast data of our own instruction manual? Thank you so much for joining me on this explainer, and remember to always keep questioning the data.